Hello, students. Very good afternoon. Yeah, give me a second. Yeah, yeah, fine, students. Yeah, hello, students. A very good afternoon. Anyways, so today we are first revision session on biological classification. So before we start the session, I would like to launch a technical poll. If my audio and video is clear, please let me know. So the poll should be on your screen. Please confirm either by message on the chat box or by the polls. Let me end the poll now. Okay, if there is any technical issues, you can be free enough to put it on the chat box and we'll make sure that it's fixed. Okay. So before we start today's session, let us go to our first question of the day. That is, give me a second. Yeah, that is, Aristotle's classification of animals was based upon what? Is it okay? Most broadest level of classification is kingdom, phylum, class, and subpole. Okay, that was the prior PPT. Level of classification is kingdom, phylum, class, and species. Okay, so these are all the topics that's already covered. So it should be very easy for you to attend these questions. So you shouldn't be taking more than 15 seconds to, answer these questions because it's pretty straight and it's straight from the textbook. It's not applicative questions. Okay. So let me end the poll. Okay, and see what's the right answer. See, the broadest level means which includes most of the common and uncommon organisms in one bigger categories. For example, if you take species, it is more narrower, and we in species we see the most similar characteristics. For example, humans. Whereas when you talk about class, class is even more broader category. In class, what will happen is, for example, you can take carnivores and herbivores, and you can put them in two different classes. But if you see in carnivore, there are so many dissimilar animals, but they are all carnivore in nature when it comes to its nutrition. So that is why carnivore is even more broader class. When it comes to phylum, for example, in phylum, we have chordata and non-chordata, right? So this is even more bigger. For example, right? So it's even more bigger. But when it comes to your kingdom carries like the most broadest of the broadest categories. Now, for example, you can have kingdom plantae, kingdom animalia, right? And then we have kingdom fungi, kingdom protista, as well as kingdom monera. So the most broadest level is kingdom organisms, right? Chordates, non-chordates. You can talk from cellular level organization to most complex organ system level of organization. So that is why A is our answer. Phylogeny refers to. So here is your next question. Phylogeny refers to taxonomy of organism, evolutionary history, evolutionary classification, and modern classification. So here are your options. So let me launch a poll now for you. Okay, please answer your questions through the poll. Okay, what does phylogeny refers to? I'm sorry. MCQ. Yeah, I think the poll question should be here. Students, I will try to fix this. Yeah, now it should be on. Yeah, the poll should be on now. So please take a little time and try to answer. Okay. Phylogeny refers to what type of classification? Chalo. We are reaching our end of time. Again, it's a direct question. Phylogeny or phylogeny is a mode of classification where we take evolutionary relationship. Okay, so basically, it's not evolutionary classification; it's evolutionary history. So, taking evolutionary history, we can really understand like where this organism could belong to. For example, if we take about snakes, if you see, we classify them under a broader group called as tetrapods. So tetrapods are the organisms that have limbs, but when you see snakes, they don't have limbs. But we still put them under tetrapods. So the reason is because when you go for the evolutionary relationships, the ancestral beings they had limbs and they lose limbs during a certain period of time during the course of evolutionary history. So that is why we choose evolutionary history as one of the major criteria to classify organisms into certain, a more reliable groups. So phylogeny is one such classification techniques where we study evolutionary history and classify the organisms based on that. Okay. So now what is classification, people? So you must be already understanding several meanings and several definitions to what is classification. See, classification is grouping organisms or grouping things into convenient groups. So that is called as classifying. Anything you group into convenience, to your convenience, that is called as classification, right? So humans have been trying to group things like on various levels, like we try to group plants into plants, animals into animals, right? So even in society, you can see we group them into several levels. In classroom, we group the students based on height, based on performance, and so many things. We always want to classify and organize things to understand better about a particular circumstance. So in our biology, the most convenient way to study all the organisms is to put them into similar and dissimilar categories. So this you know convenient, I mean, like putting the things into convenient groups is called as classification. Okay. Now, needs for classification is to understand the vastness of our biodiversity, and that is not possible to study every single individual organism separately. So that is why we need to have groups so that we can generalize the idea of what this group might contain and what characteristics they might have. Okay. So that is to put vast organisms, huge number of organisms into convenient groups and to reduce the time of studying them, and then to make the studies easier and smarter. And most probably it helps in building a relationship between one organism to another organism or one group to other groups. And now classifying has given us a broader idea that we are not like separate individuals, we are all coming from the same ancestry. Bacteria are not so much different from other organisms. We are all coming from the same background, but we have evolved or we have a different organization. That is what is the major difference, right? So that is what it is. And then it has given us a better evolutionary relationship between organisms, and it has brought the feeling of oneness, that we are all coming from the same origin. So okay. So coming to the history of classification, we know that the most earliest attempts was made by Aristotle. So Aristotle attempted one of the most scientific basis of classifying organisms. For example, he divided trees into herbs based on their height, right? So he considered that all the tree-like organisms should be belonging into trees, including fungi. He put it into trees. So he divided the trees based on their height, for example, trees, shrubs, and herbs. Whereas animals, he went on to do it even more broader way. For example, he divided animals into major categories, such as the animals with red blood and animals without the red blood. Okay, and again, the red blood and the no red blood organisms were divided For example, he found out that mammals, lizards, birds, and fishes had red blood, whereas the non-red blood usually had harder bodies, such as insects. Okay, and some of the same non-red blood organisms had soft bodies, and usually these soft bodies, some organisms had shell, and some organisms did not have shell. So in shelled organisms, he usually went on to collect the shells from the seashore or river banks, and for no shelled organisms, he saw jellyfishes and put them into one categories, right? So this is what like gives us an idea like how the origination or origin of classifying organisms have taken place from the earlier days. Now, coming to different types of classification, we have three different types of classification. One is artificial system, natural system, and phylogenetic system. Okay. Coming to artificial classification, one of the earliest people who does the artificial classification system was Carolus Linnaeus. So it is based on morphology alone. So you see the morphology of an organism and consider that whether it is a a tree, whether it is a animal, whether it is a crawling animal, whether it is a limbed animal. Okay, so something like this. Base classification based on only morphological characteristics is called as artificial classification system, and it was basically developed by Carolus Linnaeus. For example, he tried to divide plants based on vegetative characteristics as well as the characteristics of androecium, that is male gametophyte, right? So he used to divide whether the ones that have bilobed anther, or whether they are polyandrous, okay, or they are polyadelphous or monoadelphous, whether they're syncarpous or apocarpous. So based on this vegetative or sexual characteristics of the plants, he would divide them. And even though it is quite vegetative, even though we call it as vegetative characteristics, or whether it is the characteristics of an androecium, etc., it's just like physical appearance, right? You just see and visualize and make differences. So whatever this morphological based system of classification, we call it as artificial classification. Now, what is the drawbacks of artificial classification? Many organisms might look alike, but they might not be the same. For example, fungi looks like a plant, but they're not photosynthetic. Correct, right? Similarly, many of the closely related species were not, they were based on a very few characteristics. It did not go into details of what is the difference between a lion and a tiger. Why you should like consider them into different species, something like that. Now, coming to vegetative and sexual characteristics were given equal stage of determining the characteristics. Vegetative characteristics are different, sexual characteristics are different, but having them under a similar groups is not acceptable, right? So, and it is not more feasible to understand the broadness of our living system. That is why artificial classification system did not succeed to that level. Coming to the natural classification system, the natural classification system is very, very broader, because in natural classification system, a lot of criteria are taken up. For example, anatomy, physiology, morphology, ontology, cytology, biochemistry, and many other categories are taken to classify organisms into convenient groups. So natural classification system is quite evident and it is quite acceptable because it considered a lot of levels to classify an organism into a very, very convenient group. So that is why natural classification system is widely accepted way of classification system. And one of the few pioneers of developing this natural classification system is Bentham and Hooker. Okay, Bentham and Hooker are two major biologists who are responsible or who are credited for developing this natural system of classification. Now, take for example in respect to plant kingdom. In plant kingdom, we divide the plants based on morphology. First, what are those? Whether they are cryptogam, whether they are phanerogam. Okay, so cryptogams means non-seeded and seeded. Phanerogams means seeded. In cryptogam, whether they are thallophytic, bryophytic, or pteridophyta. Again, in phanerogams, whether they are naked seeds or they are closed seeds like gymnosperms or angiosperms. Again, we go to the anatomical features. So we go for the cellular structure, whether they're algal, whether they are, in Thallophyta, usually initially in the plant kingdom, they used to put this fungi under plant kingdom anyways. So in Thallophytic, we cannot differentiate the plant into true roots, true leaves, and true stems. So they started putting algae into Thallophyta and lichens into Thallophyta. Whereas coming to angiosperms, again, they divided based on the complexity of their plant anatomy. So there they divided into monocot or dicot roots, right? So this is, we consider the physiological, as well as anatomical features to divide the plant kingdom into convenient groups. So this is one example. Coming to phylogenetic classification, Hutchinson is the father of phylogeny classification. So why this is very, very essential is because today, if you observe the primates like the orangutans, gorillas, chimpanzees, and monkeys and humans, so you might not really believe that we are all coming from the same ancestry, because we are bipeds, we walk on two legs, or because we don't have fur on the body, or because our skull and our organ systems or sensory organs are quite developed. We are more analytical and more intelligent. You might have 100 reasons, but how could you prove? The only proof that could come is because of going for fossilization and fossil records, and try to build this family tree and evolutionary tree of how the life originated and how this life branched and debranched, giving rise to a lot of speciation and variation in our ecosystems that resulted in so much of diversity within an individual genus, something like this. So that is why phylogeneity is very, very important. Okay, so phylogenetic classification is more based on evolutionary history. So it gives about a timeline, it gives about modifications of organs, it talks about different categories of where we were all one, having a common ancestry, etc. So that is about phylogenetic classification. Okay, so now we talked about phylogenetic classification. Now let's go for cytological classification. Cytological classification, one of the best examples that we can pick from the cytological classification is we divide prokaryotes and organisms based on prokaryotes and eukaryotes. Why was it even necessary? Because the complexity of the cells gives rise to another story of how even the cells started evolving and becoming more advanced, right? If you see the nucleus of the bacteria and if you see the nucleus of human beings, the complexity is quite more, right? So if you see the chromosome number, chromosome size, chromosome shape, the behavior of the cells during meiosis, etc. So they're all trying to give us more and more evidence. For example, if you see in plants, we talk about sporophytic and gametophytic phases. So that's all based on cytological behaviors, right? So that is about cytology. Now, chemotaxonomy. Now, chemotaxonomy is another interesting aspect. Now, we try to divide animals based on biochemical composition. For example, we tried to divide bacteria into two domains, that is archaea and eubacteria. Why did we even do that? Even though like we put bacteria, eubacteria and archaebacteria into monorans, we were not accepting so well. To a certain stage, we thought that it is not that convenient to put these two under one group, because when we analyzed the biochemistry of these two eubacteria and archaebacteria, we came across something very astonishing, that the archaebacterial cell wall is made up of a very dissimilar type of a polysaccharide, whereas if you see the cell wall of a eubacteria, it was made up of peptidoglycan, where there was protein and glucose or polysaccharides. So it was like quite dissimilar. So that is why we understood that, oh, archaea has a different capacity to live under extreme climates, whereas the eubacteria lack this capability because of the biochemical characteristics of these two organisms. Maybe you might have come across the Taq polymerase that we use for PCR reactions. So even that is one of the biochemical proofs to tell that it is not the same protein that we have. It is something different that is protecting their genetic material and enzymes from degenerating in higher temperatures, right? So anything that you categorize these organisms or similar or dissimilarly based on the biochemical analysis is called as chemotaxonomy. Similarly, we have numerical taxonomy based on the number of hits. Like if you take the mice and if you compare to the human beings, we might get up to 70% of similar characteristics. Okay. And if you take chimpanzees and humans, you might get 98% of hits. And if you like similarly, if you consider the salivary genes of human beings and compare it to the some of the genes of amoeba, maybe you might get around 8% of the similarity or something like that. So if you analyze the genes and if you analyze the proteins, if you analyze enzymes and their characteristics, you might get similarities and dissimilarities. Based on similarities and dissimilarities, we can build a relationship again. Like, are we the same? Are we so different? Who is more similar? Who is more dissimilar, etc. So this is more of a computational analysis of similarities and dissimilarities, right? So coming to different system of classifications, we had a huge history coming to how our classification system has evolved over a period of time. Initially, it was two kingdom classification that was basically by our Linnaeus. Linnaeus divided our entire living organisms into two kingdoms, that is kingdom Plantae and kingdom Animalia. Right? Later, it came three kingdom classification where we had Protista, all unicellular was placed under Protista, all green plants, even fungi was placed under Plantae, and then we had all animal-like organisms under Animalia. Then they understood the cytological characters. All prokaryotes were put under Monera. All eukaryotic unicellular, okay, sorry, yeah, all eukaryotic unicellular was placed under Protista, and then we had Plantae, and then we had Animalia. Next came five kingdom classification system. R.H. Whitaker did an excellent job in giving a very, very foundational okay, way of classifying system where we had Monera, Protista, fungi, Plantae, and Animalia. Whereas coming to sixth kingdom, we had divided again the Monera into two domains, that is Archaea and Eubacteria, and then we had Protista, fungi, Plantae, and Animalia. So here we have the contributions. Here we have the contributions of scientists and their kingdoms on what basis they divided these living organisms and what were their limitations. See, for example, Carolus Linnaeus was the first person to give the name kingdom, and then he divided the living organisms into Plantae and Animalia, two kingdom. So here there was all photosynthetic organisms and non-photosynthetic organisms, even unicellular organisms was placed under similar kingdoms, right? So that's why it failed. Coming to Ernst and Haeckel, in 1866, they divided into three kingdoms, that is Protista, Plantae, and Animalia. All unicellular was placed under Protista. All plant-like organisms was placed under Plantae. Why I'm telling plant-like? Because even fungi was placed under Plantae. And then we had Animalia. Okay, Animalia, though even unicellular like for example, Protista was placed under Animalia. Okay, so that is our three kingdom. Next came four kingdom, that is Copeland. He understood the cytological characteristics, and then he put all prokaryotes into monerans, all eukaryotic unicellular into Protista, but he failed to differentiate the organization of fungi. He could not understand fungi very well. That's why he placed fungi into plant kingdom. That was the one limitation that was under Copeland's classification. Coming to five kingdom classification by R.H. Whitaker, he really understood that fungi are heterotrophic organisms and they are not like plants. They're completely different organisms. They behave completely different. That's why the fungi was given a separate kingdom. Now we had Monera, Protista, fungi, Plantae, and Animalia. Coming to modern classification system, we had Carlos in 1990. He taught that the Archaebacteria and Eubacteria are not at all similar, because Archaea are one of the most ancient bacteria, and their capacities, the cytological characteristics, and their biochemistry is completely different from the normal bacteria. So they have to be placed under entirely different domain itself. So that's why the entire living system was divided into three domains. One is Archaea, bacteria, and Eukarya. Three domains were given. Under Archaea, Archaebacteria was placed. All the Archaebacteria was placed under this. Under bacteria, only Eubacteria, that is true bacteria, was placed under Eubacteria. Next came Eukarya. Under Eukarya, all eukaryotic organisms such as protozoa, fungi, Plantae, and Animalia was placed. So this is about domain systems, okay, which we call it a six kingdom classification system. Now coming to how five kingdom classification system is based upon, like R.H. Whitaker, how did he conclude, or how did he even divide this classification system? If you ask the question, okay, so here is your answer. Give me a second, people. Sorry, give me a second. Yeah. Oh, people. Oh, I think my slide was. Give me a second. Oh, 1 second, students. Mm, I hope we have the slides. Okay, 1 second. Yeah, I think we have the slide. 1 second, I'll just check on the YouTube students. Okay, chalo. Hmm. Okay, people. So let's go back to where we stopped. Okay, sorry for the inconvenience. There was a problem with sharing. Anyways, yeah. So on what basis did Whitaker did classify the organisms? First one, look at this. These are very important because there can be a question coming from R.H. Whitaker's based on classification system was based on what criteria? So his criteria was basically first one cell type. Okay, in cell type, and then the composition of cell wall. Next came the nuclear membrane, body organization, and mode of nutrition. For example, if you talk about cell type, he divided the organisms whether it is prokaryotic or eukaryotic. Cell wall type, whether it is made up of cellulose, whether it is chitin, or cell wall is absent, or it is made up of polysaccharide and amino acids. Okay. Next one, coming to nuclear membrane, whether it is present or absent, that is based on prokaryotic or eukaryotic. Body organization, whether they're cellular, levels, tissue level, organ system level, or organ level or organ system level. Coming to mode of nutrition, whether they are autotrophic, in autotrophic, whether they are photoautotrophic or chemoautotrophic. Okay, and then again, coming to heterotrophic, whether they are strictly saprophytic or they're parasitic or they are carnivora. Okay, or heterotrophic, etc. So based on this, he divided this entire living organisms into 5 convenient kingdoms. So here we have a question. Carolus Linnaeus classifies the plants based on what? Okay, so here is a question, and let me launch a poll. Okay, so you have a minute to answer this question. Tell me, Carolus Linnaeus classify plants based on what? Okay, here's my student. Yeah. Come on, it's very easy. We already discussed this. Let me end the poll. Carolus Linnaeus was the person who actually introduced artificial system of classification. Give me a second. Let me go back. Yeah, artificial systemology, right? So one of the criteria he usually used to consider was the andretium. So andretium is the answer. Okay, so he took andretium. Sorry. So let's start the kingdoms. So kingdom Monera, we're going to study. So the sole members of kingdom Monera is bacteria. So bacteria are so high in number. There are trillions of bacteria in our environment. Okay, so it is very, very hard to study bacteria based on their characteristics. So that is why we have to use a certain tools to understand this bacteria. So let's see what are the characteristics of bacteria and how they are classified. Now, if you see where this bacteria can survive, monorans can survive in all the habitats that we can imagine, like very extremely high temperatures, high acidic environment, unoxygenic environments, that is anaerobic conditions, aerobic conditions, okay, in the decaying organisms, decaying organic matter, okay, as symbiotes, as parasites, okay, as saprophytes, as autotrophic, etc. They can survive in the monoron's habitat is cosmopolitan. Bacteria are very, very simple organisms, okay, but they show very complex behavior. Under Monera, bacteria are the sole members of Monera, okay. So if you see the bacteria can be classified, if they are round shaped, they are like rod shaped, they are called as bacilli. In comma shape, they are called as vibrio. Okay. So some of in bacteria, we can divide them into two categories. One is archaebacteria, other one is eubacteria. Let us generalize the bacteria. First, I told you that they can live in harsh conditions. And I told you they have a wide range of appetite. That means their buffet is too much. They can be all like phototrophic, chemoautotrophic. Okay, they can be saprophytes, parasites, symbiotes. Okay, you name it. All type of nutrition is very well explored within the bacteria. They undergo binary fission as a mode of reproduction in most of the cases. In unfavorable condition, they are known to form endospore formation. Okay, and they have motile organs such as flagella, okay, for the movement, and the body is having fimbriae and pili are the additional cell wall extensions that help in pili helps in conjugation, whereas fimbriae helps in attachment to the substance. Under bacteria, we can divide them into archaebacteria and eubacteria. Under archaebacteria, archaea means ancient bacteria. Okay, the word archaea suggests that they are very ancient bacteria. Under archaea, so if you see the ancient earth, the environment on the earth was very, very, very harsh. So in these conditions, hyper acidic or hyper temperature conditions, you can find the archaebacteria. Examples of archaebacteria are some of the salt loving bacteria such as halophiles, some of the acid loving bacteria or temperature loving bacteria called as thermoacidophiles, and some of the bacteria which live in marshy areas at the guts of organisms called as methanogens. Coming to archaebacteria, their cell wall is not made up of cellulose. It's not made up of peptidoglycan as well. They're made up of complex polysaccharides, and it can vary from one species to another species based on the conditions they are surviving in. Okay, so coming to the characteristics of one of these bacteria, let's take halophiles. Halophiles are found in some of the regions like Dead Sea. The salinity is very, very high, very, very high, where no organisms can survive basically. Here, what they do is they actually harvest sunlight based on a purple color pigment that are present in the cytoplasm that is called as bacteriorhodopsin. So this bacteriorhodopsin is a purple color pigment that is present in archaebacteria, which directly harvests the sunlight into ATP energy without the intermediate products such as glucose, okay, etc. Acidophiles are known to, oxidize some of them. They convert into H2SO4, okay, thereby this chemical energy into into ATP molecules. So they are chemoautotrophs, and they live in extremely high temperatures such as 70 to 90 degrees Celsius. Okay, and it is hyper. Okay, so for example, the Yellowstone National Park, where you have the hot springs. Okay, that is where they survive. Parthenogens are anaerobes where they live in strict anaerobic conditions or no oxygenic conditions. So they have the capacity to live in other organisms' gut. Okay, is unimaginable to even think about it. So their job is to convert the cellulose. They break down the cellulose using the cellulase enzyme that they can print, so that is methanogens. Okay, so they're helpful in biogas production. Known to be found. Okay, 1 second, people. They are known to be found in several. Sorry, people. Give me a second. In acetyl, N-acetyl muramic acid is one of the peptidoglycan that is found in cell wall of the bacteria. Okay, so they are basically motile because of the. If we talk about autotrophic bacteria, one of autotrophs. Coming to photo or blue green algae, so these cyanobacteria, they have a similar type of pigment with harvest light, okay, and observe under the microscope. They can be units. Okay, so this is the example bacterial colonies have, whereas some of these bacteria, whenever there is a lot of application and cause something called as algal blooms. Okay, so algal blooms are responsible for BOD or biological oxygen demands in lakes and ponds, which can cyanobacteria, or they're used for, are helpful in atmospheric nitrogen fixation using a specialized cells called as heterocyst. So they have special enzymes that can convert this atmospheric nitrogen into nitrites, nitrites, and ammonia, which is very useful forms of nitrogen for our to break down the chemical energy for the production of nostoc and anabena. So the cyanobacteria, they convert this inorganic nitrogenous substances such as nitrates, nitrates, and ammonia, okay, into useful form, and they produce ATP. They're called as nitrifying nitrogenous products and phosphorus, iron, and stuff. very important nutrients for plant growth and development. Sorry, students, there was a network problem. Give me a second. Let me share back the screen. Yeah, current slide. So let's continue. Okay, so coming to heterotrophic bacteria, as I was saying, so some of them can be really a big problem to us. For example, if you take tetanus, tetanus was responsible for killing millions of soldiers that were wounded in the World War I. So such kind of diseases, cholera killed so many babies back in the 90s when we didn't have the proper antibiotics. Okay, and even today we can see that some of the brownish spots that we can find in the lemon, orange, or sweet lime. So this is because of the citrus canker. Okay, that is the bacteria that is creating the disease. Okay, so we know that bacteria can reproduce by one of the major asexual type of reproduction, that is binary fission. Okay, under unfavorable conditions they are known to form endospore formation. Okay, and these endospores help them to survive in extreme conditions. Okay, and whenever there is a need for unfavorable conditions, they undergo a type of sexual reproduction which is called as pseudocopulation. Here it is called as conjugation, where the characteristics are exchanged between the two different types of bacteria. Okay, here when you talk about the genetic material, we know that the genetic material of a bacteria is present in the cytoplasm, and it is not having any nuclear membrane, and the DNA is present as nucleoid. So double-stranded circular DNA or double-stranded chromosome is present in the bacteria, whereas outside the nucleus there is also double-stranded circular DNA that is called as plasmid DNA. Okay, so plasmid DNA is present, and this plasmid DNA is very responsible for the bacteria's characteristics, such as, for example, it gives them resistance to antibiotics, fertility factor, or cholecinogenic factor. All of these factors are coming from your plasmid DNA. Okay, if anything is asking for what is plasmid DNA, plasmid DNA is also called as extra chromosomal DNA. Okay, so chalo. So one of the smallest known bacteria is Mycoplasma. Mycoplasma is the smallest known bacteria, and this Mycoplasma does not have any cell wall. They're highly resistant to antibiotics, and they're very problematic organisms because they can cause severe diseases like tuberculosis, intestinal or lung TB, and they are also known to infect the patients with burnt wounds. That's why healing the patients with burnt wounds is very hard. Okay, so let's go for the next question. Which one of the following are the examples of eubacteria? So it's an easy question, but a tricky one. I think you can, okay, relaunch the poll. Okay, let me relaunch the poll. So the poll is live, people. So your options are Paramecium caudatum, Escherichia coli, Euglena viridis, Amoeba proteus. Okay, so these are your options. It's a 2011 question. Should be so simple for you to answer. Which one of the following is not the example of eukaryotic cell? Okay. Right? Did you get the answer right? Chalo. I will end the quiz because it's an easy question. See, people, euglena or euglenoids, when we talk about euglenoids, when we talk about protozoans, we come across Paramecium, we come across Euglena. Okay, they're all belong to Protista, isn't it? And then we have Amoeba, Euglena, Paramecium. They're all belonging to protists, right? When it comes to Escherichia coli, even though you don't remember Escherichia coli, okay, like round shaped cocci, okay, and then rod shaped bacilli, okay, coli, which is a round shaped bacteria. They are called as what? Are they eubacteria? Are they eukaryotes? Are they prokaryotes? So bacteria, all the bacteria belong to prokaryotes, right? So the Escherichia coli is a prokaryote and do not belong to the group of eukaryotes. That is why option is B, which is not matching with the rest of the group. And Paramecia, are all belonging to option is legit. Okay. So students, so we will end this session here, and from the next session we'll start with Protista, okay, general characteristics and main characteristics of Protista, and followed by fungi, and we can probably conclude the revision session tomorrow. Okay. So we had some technical difficulties today, so probably we don't have the same thing tomorrow. Thank you, students. Thank you for attending the session. I'll see you in tomorrow's revision class. Have a nice day. Take care. Bye-bye.